Hello, biology superstars. I know that you are all starting to study for your IB exams. So I wanted to make some tricks and tips videos. This one is going to focus on how to tackle the data-based questions. So let's get started. As an overview of the external assessments that IB presents you with, you will sit for two different papers. Remember, it says here that these are worth 80% of your overall score because your internal assessment, your IA, is the other 20%. That should be very much done and submitted to the IB by now. So what you're focusing on as you're studying is the paper 1 and paper 2. We'll go through SL first because the requirements for these papers are a little bit different. So SL, you will sit at paper 1 that is an hour and 30 minutes and it has two parts, multiple choice questions and database questions. At the SL level, there are 30 multiple choice questions and 25 points worth of database questions for a total of 55 marks. And that is worth 36% of the 80% of your score that comes from these external assessments. For your paper 2, you will have an hour and 30 minutes to complete the two sections of the paper 2. Section A is 34 points of data-based and short answer questions, and section B is 16 points of extended response questions, and that is a total of 50 points. And that 50 points is worth 44% of your grade for the external assessments. For HL, you guys have 2 hours for your paper 2. That is because you have more content to cover and more questions to answer. You have 40 points of multiple choice questions, and the remaining 35 points come from the database questions for a total of 75 points. The percentages are the same. That's 36% of your overall score. The paper 2 is also longer, 2.5 hours. For you guys, the section A database and short answer questions are worth 48 points, and the section B extended response is worth 32 points. That's because you do an extra extended response prompt. All right, so I don't know if any of that is big news to anybody, but I do think it's just helpful to see all of that laid out about what this means for your exams, and sort of how this part of the assessments for the IB works. So let's focus on this part 1B of the paper 1. We'll go, so database questions. They are exactly what they sound like. They provide you with some type of data, usually in the form of a microscope image, a graph or a table, a kite diagram, or a bar and whiskers plot, or an enzyme graph, and then they ask you questions about that content. They could be skill-related questions, so it could be stuff that's like, how do you deal with the skills that are associated with looking at this image, or it could be content-related questions. And we'll go through some examples, looking through some images right now. My writing tools are acting a little bit funny, but I'll just see if they sort of cheer up as we go through this a little bit more. So, for example, there's my pen. This is obviously a microscope image. And remember that when you're looking at a microscope image, the very first thing that pops in your mind should be, oh, they are going to ask me to do some type of a calculation that has to do with the magnification, the image, and the actual size of whatever it is that I am looking at. So you should be ready to at least attempt some type of a calculation. Based on this triangle of calculations, you should also always remember the conversions between micrometers, nanometers, millimeters, and centimeters or meters. So I'll write it up here. Remember that in 1 micrometer, there are 1,000 nanometers equals 1,000 nanometers. So nanometers are our smallest thing. And then in 1 millimeter, there are 1,000 micrometers. So 1 millimeter, and I'm working really hard to make my m's different here, is equal to 1,000 micrometers, and the micrometer is the m with that funny little tail at the end of it. And then in 1 meter, there are 1,000 millimeters. That hopefully is not like big news to anybody. But I'll write it just in case. So 1 meter equals 1,000 millimeters. And you might also sometimes have to convert between centimeters and millimeters. In 1 centimeter, there are 10 millimeters. All right, so all of these conversions come into play when you're looking at an image like this. A typical question that they might ask you is like, they could give you a scale bar and say that this scale bar is 2 centimeters big. 2 centimeters big. Like the actual measured size of this with a ruler is 2 centimeters. And that it represents, so this actual thing is 2 centimeters. The distance that it represents is 10 micrometers. 10 micrometers. And they want you to calculate what is the magnification of the image. So if you're doing that, the scale bar is the image or drawing that you are looking at. So it would be 2 centimeters over the actual size of the thing, which is 10 micrometers. Remember, they told you that. 10 micrometers. And I'm going to make sure that my M has the funny little tail. And then you need to calculate the magnification. Before you calculate the magnification, you need to get these two sets of units in to match. So I will do it probably by getting centimeters down into micrometers. So 2 centimeters is 20 millimeters, and 20 millimeters, to get between millimeters and micrometers, we go here. And 20 millimeters is 20,000 micrometers. So the conversion would be, I'm going to do it over here because I'm running out of space, 20,000, 1, 2, 3, divided by 10. And the magnification that you are looking at when you cross out the zeros is 2,000. And remember, magnification does not have units. So that is like one example of a question that they will almost definitely ask you if you are looking at a microscope picture. And then it gets into like other skill related and content related stuff. So generally, I'm going to clear this just to give some more space. Generally, I would say that there is like 6 to 10 points of questions for each of these sets of data that they're asking you about. And it could be like, in this case, it could be content related, like what types of cells are these? How do you know? Describe, it could be skill related, like describe the mounting technique of how to get these under a microscope. So just be, as you're going through the curriculum and studying, be looking for things that are represented with diagrams in your textbook, and keep an open mind to the kinds of questions they could ask you. We'll run through a few more examples, but it really is like the whole idea is that they can ask you anything. So we'll talk about that a little bit more at the end. All right, so another example of a type of question they could ask you is about a kite diagram or a box and whiskers plot or a graph and table. Here we're looking at a graph and table with Simpson's diversity index. So they're giving you 3 different types of forest ecosystems. They're just calling them A, B, and C, and giving you the Simpson's diversity index values. If you need a review of Simpson's diversity index stuff, that is in A4.2 or like widely available on the internet. Go over it a little bit. I would know Simpson's. I would also know Lincoln's, which is the like how you estimate a population size. And you don't need to know how to do these things, but you need to know how to recognize when they are being done and talk about data that is produced by them. So here, for example, they could ask you like content related stuff. What is the difference between these ecosystems that might result in these different index values? Skill related. What is the difference between these numbers? Like which one's good and which one's bad? Remember, the higher the Simpson diversity index, the more evenness the populations that are present in a given environment show. So you would need to sort of be able to give basic information about what they're showing you. And also they might ask you to like evaluate, like evaluate the study that might have produced this data. And then you would say like, well, we don't actually know very much about the study that produced this data. We don't know how big these areas were. We don't know what the sample size was. We don't know what variables were controlled. We don't know what variables were not controlled. We don't know how populations were measured. So if they're ever asking you to evaluate, you need to give pros and cons to like whatever scenario they're presenting you with. Pros and cons. All right. So we'll do, I think, just one more example. I'm going to finish writing cons. We'll do, I think, just one more example for a type of question that they could ask you. Here is a box and whisker diagram. If you need to review box and whisker diagrams, go to, where's my pen? Go to D3.2 and specifically D3.2.15. And I think most of my IB students seem like this has also come up in math. I'm going to just try to erase this like extra little stuff that's over here before we get started with this. All right, so box and whisker diagrams, they're telling you a few things in the data, and you should be able to comfortably talk about like what is going on with this data set, where the first quartile, second quartile, and third quartile values are in a box and whiskers plot that the IB would show you. And I think in all of them I can think of, the median value is the same as the second quartile. And the extreme values are outside of the quartiles. And remember, an outlier, if there was an outlier in this data set, would usually be shown with like a little star off to the side. So they could show you, I think in the syllabus, the box and whiskers example they use is like for the continuous variable of height in a classroom. They could show you one about height. They could show you one about anything and say like, what is this data showing you? What is the median value here? Estimate a median value. And then you need to like draw this line down to here and say, probably 29. And if you're asked to estimate, they'll usually accept a range of answers. Here it would be like between 29 and 30, I would imagine. And that's all skill related stuff that you're like doing with a ruler, looking at the data. Content related stuff, if they're asking you about height, they could ask you to like describe what continuous versus discrete variables are. So be ready for anything in these database questions. Like remember that it can be based on content as well as skills. You would do well probably to go through the tools section of the IB syllabus. Because in the tools section is where they will tell you, I'll write it, tools section. I think it's like in the pages in the 30s somewhere in syllabus. And that's where they tell you like what mathematical what mathematical equations they expect you to be familiar with, and what mathematical processes they expect you to be able to do and recognize, what types of like scientific lab work they expect you to be sort of familiar with. So like all of this stuff is in there. So as you're studying, it'd be worth it to flip through that tools section of the syllabus for sure. And let's go on to the last 5 things to think about as you're going over how to study for these DBQs. So do lots of practice questions, because you'll notice stuff that comes up more than one time in the practice questions, like that your teacher gives you, or Save My Exams is great. Any IB review subscription review website will have practice questions for you to do. This is the time to sign up for those websites. Do a 1-month subscription. Do it now. When you're taking the exam, make sure that you are keeping an eye on your time. You will get the booklets for paper 1A and 1B together, and then have whatever your total allotted amount of time is to complete both of them. So you can like do whichever one you want first. You can go back and forth between the two of them, but make sure that you're keeping an eye on how much time you have left. I do the easy questions. What I would do is flip through both of them, do the easy questions first, the ones you know, build your confidence, and then go back to the ones that you're struggling with a little bit more. Especially in this DBQ section, check the point values. It seems like most of these questions are ranging between 1 and 4 points. Really, on the SL exams, I haven't seen any that go above 3 points. So don't waste a bunch of time like beating yourself up over a 1-point question. Just keep moving and come back to it if you have time. Review the command terms. So like that idea of if they're asking you to evaluate something versus asking you to describe it, they're asking you to do very different things. Know what those command terms mean. Read the prompts super carefully. I always say like read them twice and underline stuff in the prompts, because there might be like one tricky little word that you don't notice. Like, are they asking you about eukaryotic or prokaryotic cells? That's important. You would write different stuff. So spend that extra 10 seconds really looking over the question and making sure you understand what they're asking you to do. And then especially in the DBQs, get the little stuff points. Make sure you're always showing your units, showing your work and your calculations, adding a little bit more detail than you think you need, because you never know what's going to be in that mark scheme. And as long as the things that you are writing down don't directly contradict each other, they might count towards a point. All right, so this is it for DBQ review. I hope this has been helpful, and good luck on your exams.